Well, we've looked at how little atoms can come together and form life's building blocks like amino acids and the letters of DNA and RNA. And we've looked at how those building blocks can form things like proteins. We can go up one more level to how proteins come together to form molecular assembly lines. And that's what I want to talk about today. It's actually interesting to think about molecular assembly lines. So before we delve into the details, why don't we step back and say, well, what are assembly lines? And think about Henry Ford himself and the revolution he caused when he brought assembly lines into the automobile industry. So what exactly was the revolutionary part of Henry Ford's idea? Can you think about what it was that he did that nobody else did before him? What was manufacturing like before Henry Ford? And what exactly did he do that brought advantages? What were the things that he did that were different from other people? Also, you might want to think about how Henry Ford's ideas in the auto industry impacted the rest of society. You may have read a book, The Brave New World. Brave New World. People were thinking about these ideas very early on in the time after Henry Ford. So I don't know if Henry Ford looked at biology very much, but if he had lived in today's day, he would have gotten lots of great ideas from inside the cell that he couldn't have possibly known back when he came up with his ideas about assembly lines. Because it turns out that proteins work in assembly lines. They really do. They work in sequence, one doing one job, one doing the next, the one doing the next. In some cases, transcription and translation can be like this. For example, the RNA polymerase is what does the job of transcribing DNA into a special kind of RNA called messenger RNA or mRNA. So you have this machine, this protein machine called RNA polymerase, doing the job of copying DNA into RNA. The RNA that is produced gets immediately fed into the machine called the ribosome that we talked about last time, that is in charge of translating the four-letter code of RNA into protein. So that's the next step down in this assembly line. It feeds into the ribosome and starts producing protein. Now what's interesting is that it's not just one ribosome that grabs onto one mRNA. It can be several ribosomes that grab onto the same mRNA. Just like on an assembly line, there can be several things happening. And the tail of the mRNA can be going out of one ribosome into another ribosome that then makes the same type of protein right there on the end. And this can happen over and over again so that multiple copies of the same protein are produced before the RNA is even fully out. Those proteins are all coming out of the ribosome. So it's a fascinating way of speeding up things inside of the cell. Now, we can understand a little bit more about these assembly lines because just like in Ford's idea, you have all these things happening sequentially. So a story that probably can help you in this regard is the story of a man named Bruce Alberts, who is well known for his cell biology textbook and was also the National Academy of Science president back in 1999. And at that time, he discussed a new view of the cell. He said that we shouldn't think of cells as bags of chemical reactions. Instead, we should think of cells as full of molecular assembly lines. The message was that we shouldn't underestimate the complexity inside of the cell. Now, many people had been thinking about that for a while. He wasn't the first one. But probably a lot of people who were teaching in the school system still just basically thought of the cell as a bag with a few organelles that were in charge of different things and reactions just sort of happening in there the way you might imagine them happening inside of the bottom of a test tube as you shake it or put it over a Bunsen burner. But no, he said, think of it as inside of the cell, there's little regions that cellular processes are going on in sequence along a molecular assembly line. in previous years, people thought of the cells not even as a bag of chemical reactions, but as just a glob of protoplasm, kind of like a little lump of jello with nothing really happening in there, just as building blocks for other things. So it's not surprising that 100 years after people thought of them as globs of protoplasm, we're now having to think of them as something even more complex. And 100 years from now, we might think of them as even yet another level more complex than what we think of them as now. So these are the kinds of things that you realize as you start to understand the hierarchy of building up life, from atoms to molecular machines to molecular assembly lines, all the way up. And if you're going a little bit deeper, you can now look at the leaf that deals with molecular machines and their assembly lines, and how that all fits together.